Imagine turning ordinary bacteria into microscopic factories. In this video, you'll learn bacterial transformation step by step. Subscribe, and let's get started. Can scientists really teach bacteria new tricks? Absolutely! Let's discover the amazing process called bacterial transformation. Bacterial transformation is the process of introducing foreign DNA into bacteria, allowing them to copy and express new genes. Scientists use transformed bacteria to produce medicines, research proteins, vaccines, enzymes, and many biotechnology products. Normally, bacteria don't easily accept foreign DNA. First, scientists must prepare competent cells. These are special bacterial cells that are ready to take up DNA. The first method is chemical transformation. One of the simplest and most widely used laboratory techniques. Calcium ions reduce the electrical repulsion between the negatively charged DNA and the bacterial surface. With less repulsion, plasmid DNA can move much closer to the bacterial membrane. Next comes the famous heat shock, where cells are briefly warmed to about 42 degrees Celsius. The sudden temperature change temporarily increases membrane permeability, allowing plasmid DNA to enter the cell. Congratulations! The bacterium is now successfully transformed with the new plasmid DNA.
If you enjoyed this video, like, subscribe, and turn on notifications for more exciting molecular biology adventures.